Hello students, today you are going to study three laws of Mendel. So the first law of Mendel is law of dominance. It is a first law of inheritance. In a cross between the two pure parents for any pair of contrasting character. The character which appears in F1 generation is called as dominant one and the other one is called as recessive one. So, as I said earlier, according Mendel, each character is controlled by a pair of factors or the genes. And when it is in pair of dissimilar factors, only one factor expresses its trait in F1 generation, which is called as dominant one, and the other one is called as a recessive one. Let us see this law of dominance with the help of monohybrid cross. Yeah. So this is a cross between the tall plant and the dwarf plant. So when this tall plant is crossed with the dwarf plant, so this tall plant possessing the genotype, that is the dominancy and the dominancy, and this one is recessivity and the recessivity. So this will be the first gamete and this will be the first gamete for this particular plant. When this gamete fuses with this one, so the F1 plant will be the dominant, uh, F1 plant will be the tall plant, but this tall plant is heterozygous tall. It is not a homozygous tall, but this one is homozygous tall. This is homozygous recessive plant, and this is homozygous dominant one. Okay, this is homozygous recessive, this is homozygous dominant, while this one is heterozygous tall plant, heterozygous. So as I said earlier, in this F1 generation, this T is dominant over this one. So this T expresses its trait in F1 generation, and this T does not express its trait in presence of this one. So this is called as a recessive and this one is called as a dominant. So this is all about the law of dominance. So this is the first law of dominance. This law can be uh, explained with the help of a monohybrid cross. Let us see the second law of Mendel, that is the law of segregation or law of purity of gamete. Yes, this is second law of segregation. Law of segregation. It is the second law of inheritance. Members of allelomorphic pair in a hybrid remain together without mixing with each other and separate or segregate during the gamete formation. Thus, the each gamete receives only one out of two factors and are pure for that given trait. Let us see this particular law of segregation again with the help of the monohybrid cross. So these factors remain together without mixing, and while gamete formation they produce separate gametes, and each gamete receives only one factor. So they do not mix with each other, they get separate from each other during the gamete formation. That's what related to the law of segregation. And this particular gamete, each gamete is pure for that particular trait. So this particular law is also called as law of purity of gamete. So this law can be explained with the help of the monohybrid cross. This law is universally applicable. Okay, so this is simply regarding the second law of Mendel, that is law of segregation. Let us see one more law, that is the law of independent assortment. With the help of the dihybrid cross, you can see this law of independent assortment. This is the third law of inheritance. This law can be explained with the help of the dihybrid cross. When the two homologous parents differing in two pairs of contrasting traits are crossed with each other, the inheritance of one pair is independent of the other. So the inheritance of one pair is independent of the other. In other words, when a dihybrid forms a gamete, assortment or the distribution of the alleles of different traits is independent of the other. Let us see this dihybrid uh, third law of Mendel, that is law of independent assortment with the help of the dihybrid cross. So this is the This is first parent and this one is second parent, but this considering two characters simultaneously, so this is dihybrid cross. So these are two factors for the round seeds and these are two factors for the yellow seeds. Okay, so during gamete formation they do not mix with each other and they produce separate gametes. So the distribution of this particular, yeah, as I said earlier, when the two homologous parents, when the two homologous parents differing in two pairs of contrasting traits are crossed with each other. Now these two parents are crossed with each other considering two characters or differing in two characters. The inheritance of one pair is independent of the other. So the inheritance of one is independent of the other. In this is for round and this is for the yellow one. Okay. In other words, when a hybrid forms a gamete assortment, this is simply regarding the in hybrid, dihybrid forms gametes. This dihybrid forms gametes. The gamete assortment or the distribution of the alleles of different traits is independent of their original one. So during gamete formation, okay, in this dihybrid cross, during gamete formation, uh, the distribution of the gametes, the distribution of factors in gametes is independent of the original one. So in other words, when dihybrid forms, gametes assortment or the distribution of the alleles of the different traits is independent of their original one. So this is simply regarding the third law of independent assortment. Okay, let us move towards the one more important point, that is the back cross and the test cross. So back cross is nothing but it is a cross between the F1 progeny and any one parent is called as the back cross. Okay, I repeat my sentence. It is a cross between the F1 progeny of the monohybrid cross with it is a cross between the F1 progeny with a any one parent is called as a back cross. Let us move towards the one more cross, that is the test cross. In test cross can be defined as it is a cross between the F1 progeny and the recessive parent only is called as a test cross. So test cross is basically back cross. Test cross is a definitely a back cross. Okay, but that back cross involves the recessive parent and the F1 progeny. So test cross, again I repeat, test cross, it is a cross between the F1 progeny and the recessive parent. So this is used to test whether the offspring is homozygous pure or the heterozygous pure. Let us see this cross with the help of a example. So this is F1 progeny and this F1 progeny is crossed with the recessive parent. So these are the two gametes of the recessive parent and these are the two gametes of the F1 progeny. So when this gamete fuses with this one, this will produce the violet colored flower. Then this one again the violet colored. Then this one will be the white. This one will be the white. Okay. So this is simply regarding the test cross. So test cross can be defined as it is a cross between the F1 progeny with the recessive parent. Now just move towards the most important part of the Mendelian inheritance, that is the neo-Mendelism or the post-Mendelism inheritance. Okay, after the discovery of the Mendel, many scientists performed experiments on the different plants and the animals. Many exceptions or the deviations to the Mendel's ratio, different ratios were observed. On the basis of these observations, different patterns of inheritance or the gene interaction were discovered. So these deviations are called as post-Mendelism or the post-Mendelian inheritance. Okay, so this is also called as neo-Mendelism. Now just see the example, that is the incomplete dominance. Yeah, this is incomplete dominance. So first, post Mendelian concept, that is the incomplete dominance. In incomplete dominance, both the types of genes, both the genes of allelomorphic pair. I repeat my sentence. In incomplete dominance, both the genes of allelomorphic pair express themselves partially. One gene cannot suppresses the characters of the other one completely. Thus, in this, no one is dominant and no one is recessive. Okay, so the with the help of example, you can see this particular concept, that is the incomplete dominance. So this is uh, red color flowering plant, that is 4 o'clock plant. The common name of this particular plant is 4 o'clock plant, or the scientific name of this particular plant is Mirabilis jalapa. So when red color flowering plant is crossed with the white coloring flowering plant, Mirabilis jalapa is crossed with this one, red one. So this is a genotype of for the red flowering plant, and this is a genotype of the white coloring flowering plant. When the red flowering plant is crossed with the white flowering plant, uh, this plant will produce the gamete, that is the R and capital R and the capital R, and this one will produce the R and the R. Then when this one fuses with this one, the F1 generation will be the pink flowering plant. Okay, so as I said earlier, so in this F1 generation, no one is dominant over other one. So both these genes, or both the allelomorphic pair, express themselves partially. So they produce the intermediate character, that is the intermediate color, that is the pink color. So these, after crossing between these two, they will produce a pink color flowering plant in the F1 generation. Okay, and the F2 ratio, when this plant is self with each other, this will produce, in this example, when F1 progeny was self to raise the F2 generation, the F2 ratio is 1 as to 2 as to 1. That is, yes, red as to pink as to white. So the ratio is 1 as to 2 as to 1. So this is simply regarding the incomplete dominance. Let us move towards the one more concept, that is the let us move towards the one more concept, that is the codominance. Codominance, yes. In codominance, both the genes in the allelomorphic pair express themselves equally in the F1 hybrid. A classical example of this particular cross is a skin coat color in the cattle. So there are two types of cattle, two types of skin coat color found in the cattle. First one is red coat color, and the second one is white coat color. When this red coat color cattle is crossed with the white one, so they will produce the F1 progeny, and that F1 progeny will be the roan. Roan means it is a mixture of the red and the white. As I said earlier, in codominance, both the genes in the allelomorphic pair express themselves equally. So the roan is a mixture of the red and the white. So it is a classical example of the codominance. Okay, codominance. So there are two types of the, as I said earlier, coat colors found in the cattle's. So red coat color and the white coat color. So roan is a mixture. F1 offspring will be the roan. So roan is a mixture of red and the white. So when F1 was self to get or to raise F2 generation, the F2 ratio showed red as to roan as to white. That is again 1 as to 2 as to 1. So red progeny in F2 generation, the red progeny will be 1. Then there will be 2 roan and 1 white. So the red as to roan as to white ratio is 1 as to 2 as to 1. Means 1 red, 2 roan and 1 white. Let us move towards one more example, that is the multiple alleles. Two or more alternative forms of a gene in a population occupying the same locus on a chromosome. Locus means the loci or the position, are known as the multiple allele. So the classical example of the multiple allele is Drosophila, wing pattern in the Drosophila. One more example of the multiple allele is blood group in the human being. Let us see, multiple alleles arise by the mutation of the wild type gene. Okay, so mutation means the sudden change in the original gene is called as a mutation. So a gene can mutate for a several times, producing series of alternative expressions. In these alleles, they show different types of patterns: dominant recessive interaction or the relation codominance, as well as the incomplete dominance. In Drosophila, there is a series of alleles for a wing pattern, ranging from the normal wings to the no wings. Let us see the example of the Drosophila. Yes, this is a classical example of the multiple allele. So this particular Drosophila shows different types of wings, and the genes responsible for this pattern is VG plus for the normal wing, then the VGNL for the nicked wings, then the VG. These are multiple alleles of the wild type, original one. Okay, after mutation. Okay, then the VGNO, that is the notched, then the strap wing, that is VG, then the vestigial VG. Okay, so this is simply regarding the different wing patterns found in the Drosophila. So these are different genes or the mutant genes of the wild one. So more than two alternative forms of a gene is called as a multiple allele. So this is a classical example of the multiple allele, that is the wing pattern in the Drosophila. One more example is there, as I said earlier, that is the blood group in the human being. Let us move towards the one more thing. Let us move towards one more example, that is, yeah, pleiotrophy. So pleiotrophy is simply regarding the a single gene controls many traits. So let us see what is pleiotropy. When a single gene controls two or more different traits is called as pleiotropy, and the phenomenon is known as a pleiotropism. Okay, and the ratio is 2 as to 1 instead of 3 as to 1. Okay, now just see the pleiotropism. So the classical example of the pleiotropism or the pleiotropy is sickle cell anemia in the human being, and the gene responsible for this is HBS. Okay, the gene responsible for sickle cell anemia is HBS, and the healthy gene is HBA. Let us see the example of this pleiotropy. Okay, uh, so sickle cell in sickle cell anemia, RBC, our RBC becomes sickle cell or the half moon shape due to the deficiency of the oxygen. Okay, what happens actually in this particular uh, cross, when the carrier female marriages with the carrier male? Okay, so the gene responsible for sickle cell anemia is HBS, and the healthy gene is HBA. Now this is carrier, and this one is also male is also carrier, and the female is also carrier, and the genotype for this female is HBA and the HBS, and the genotype of this male is HBA and the HBS. When this female marriages with this male, okay, they will produce gametes, and then they will produce the offsprings. Okay, so this will produce the gametes like the HBA and the HBS. Then this will produce the gametes like the HBA and the HBS. When this gamete fuses with this male gamete, so the F1 offspring will be HBA and the HBA. Okay, then the second, when this second gamete, first gamete fuses with the second gamete of the male, that is the HBA and the HBS. So second offspring be the will be with the genetic makeup, that is the HBA and the HBS. So this will be definitely a normal one. Okay, that is This is possessing the genes responsible for the uh, sickle cell anemia. Sorry, I repeat, this offspring possessing the genes, that is the HBA and the HBA, and both genes are normal. Both genes are normal, so this will be the normal offspring, and this will be the carrier HBA and the HBS, because out of these two genes, one gene is responsible for the sickle cell anemia. So this offspring is possessing the HBS. One gene is HBS responsible for sickle cell anemia, and this one is normal gene. So this offspring will show a mild symptoms of the sickle cell anemia. So this is called as a carrier one. This will be also the carrier one, that is the HBA and the HBS. Okay, so this is also the carrier one. Okay, and this shows the mild symptoms of the sickle cell anemia. Now this is simply regarding the last combination when this gamete fuses with this gamete of the male, that is HBS and the HBS. This will so when this gamete fuses with this one, they will produce the offspring, that is with the HBS and the HBS. Now the both genes are responsible for the sickle cell anemia. Both genes are HBS. It means that this one will be the total anemic. It means that this will dies. Okay, so the final ratio is, instead of 3 to 1, the ratio is 2 to 1. That is the two offsprings will be carrier and one offspring will be a normal one, and this last one dies due to the anemia. Okay, so this is simply regarding the classical example of the sickle cell anemia, in which when a female carrier female marriages with the carrier male, they will produce three different types of the offsprings. The first one will be the normal, second one will be the carrier one, third one will be the carrier one, and the last one will be the total anemic, which will die due to the anemia. So this is simply regarding the sickle cell anemia and the ratio of the sickle cell anemia example. Okay, this is a classical example of the pleiotrope, in which a single gene controls many traits. In this example, this gene, that is HBS, in this combination in carrier, this behaves like the carrier, and in this homozygous condition, in this heterozygous condition, this becomes a carrier, and in homozygous condition, this is heterozygous. Why this is heterozygous condition? Because there are two genes, no, HBS and the HBA. So this is heterozygous condition, this is heterozygous condition, this is homozygous condition for healthy gene, that is HBA and HBA. This is heterozygous condition, involves HBS and the HBA, two genes, and in this also there are two genes involving in this combination, that is HBS and the HBA. Okay, so both these are carriers, and this is also this one is homozygous condition. In the homozygous condition, this gene becomes lethal, and this causes the death in that particular children. Sorry, this particular child. This is one, no. So this causes a death in the child. So this is simply regarding the behavior of the gene. Single gene behaves two ways. Uh, that is a single gene, when a single gene controls two traits. In this condition, in homozygous condition, this behaves like the lethal one, and in this heterozygous condition, in homozygous condition, this behaves like the uh, lethal one, and in this heterozygous condition, this behaves like the carrier one. In this way, this particular gene controls two traits. That is, in heterozygous condition, it becomes carrier one, and in homozygous condition, it behaves like the lethal one, which causes the death in the child. So the ratio of this particular example of this particular cross is 2s to 1. That is, two carriers and one is normal, and this dies due to the sickle cell anemia. So the final ratio is 2 as to 1 instead of 3 as to 1. So this is simply regarding the pleiotropic. Thank you. Remaining part we will see later on in next lecture.